All right. Hello and welcome, everyone. And welcome to you, Han.
Hi, Trym. Thanks for having me on the show.
How's it going?
Very good, thank you.
How do you like the event so far?
I think it's pretty great.
Yeah?
Great community. Mind you, this is my first time to an actual blockchain event.
Is it?
I'm
Wow, okay.
I'm kind of more on the from an outsider's perspective.
Yeah.
Mostly operate more in the deep tech, so machine learning, AI community, the medical sciences. So it's really great to go outside of that ecosystem and see what else is out there.
Yeah, and it's so great to get people outside as well that aren't, like fully into the blockchain thing. But
Yes.
just kind of like experiencing all of this for the first time. It's pretty cool. So
Yep.
You're joining us here from Cortical Labs,
Yes.
and we have a little something here on the table between us, and you've been demoing it outside. Why don't you tell me a little bit about what this thing is?
Yeah, absolutely. I mean, I guess this is now your CL1 device, but let me walk the audience through. Essentially, what we have in front of us, and for those of you who can't see it, the CL1 device is the product that we have been developing at Cortical Labs since the breakthrough paper in 2022 in the journal Neuron, where we showed that dish brains, when stimulated in the right way with a incentive and disincentive signal, can learn to control virtual elements in simulation programs like the game of Pong into a product that you can not only have a dish brain, but additional hardware to interface with the chip and the neurons for reading the electrical activity, for providing stimulus, but also a complete life support system, because these neurons need to be maintained at very strict physiological conditions that replicate what the human body has. So the CL1 is, I guess you could think of it like a very large shoebox-sized device. For those of you who know much about data centers and computing hardware, there are actually 3U rack-mountable sleds. So 3U being the 3 units of height for a server rack. So if you had a, say, 30U, you can potentially pack 10 of these, 10 shelves worth of CL1 units. And in the front portion, we have we can actually open up the lid first, and we can show you that the front portion is where we load the dish brains. So here is one that we have. We doesn't actually have any neurons here, but we've been demoing with some fluid to simulate what it would look like. And this is a multi-electrode array device, aka the dish brain.
This is wild. So
Yeah.
it's like this glass little thing with a circular, I don't know, like tube in the middle, and there's a bunch of like plastic tubes coming out of it. It's like full of red liquid at the moment, and there's like the little chamber that it comes into. It's pretty freaky looking, honestly. It has like all of these spiky looking things.
Yeah, certainly. So what you're describing here is so the multi-electrode array is this glass petri dish that has a well. It can contain about 5 mils of fluid, and this is fluid that keeps the cells alive. Cell culture media that kind of replicates cerebral spinal fluid, which is what bathes our brain in our skulls. And then we cap it with a stainless steel metal cap with a glass window in the middle, so we can actually still do imaging. So we can put it under a microscope. We can see what the cells are looking like, where they're growing in regions that have electrodes or not. And then we have ports here. One that's marked with an I, and one that's marked with an O. I guess it's part of your branding, right?
Input,
output. And it's actually it's exactly that, input, output, where we can input the clean fluid, and then we can output the dirty fluid, right? Because as cells compute or as they function, they metabolize, they take nutrients in, they produce waste. So this whole system here gives us the ability to pipe in fresh media, which will have high levels of oxygen, high levels of glucose, and remove waste materials such as ammonia, which is the primary component of our pee, really, and lactic acid, right?
Because so that actually comes directly from the cells.
Correct, yes.
All right, okay.
So ammonia is a byproduct of just protein degradation, cellular metabolism, they literally just excrete it.
That's pretty wild, because the whole instrument here, there's pumps and there's like these containers of fluid and these filters and some gas and everything, and it's basically just this body that's kind of there to sustain the neurons.
Yeah, absolutely. I think, we looked at it and we said, well, not only do these neurons have to maintain strict physiological conditions, because they don't actually have an immune system like we do. They're very susceptible for contamination from the environment. So the environment's actually full of contaminants. Like you have viruses that go around. You have things that we don't think much about, like mold and fungus.
Yeah.
Mold primarily actually is very common now.
Yeah.
We have a very good way of dealing with it with a very strong immune system. But these cell cultures don't have any they don't have any leukocytes. Which are white blood cells. They don't have any T cells or B cells. So what we end up doing is we end up creating this closed loop circuit with the fluid that maintains internal sterility. But we use these filtration units as a way of exchanging the waste material for the nutrients. But having a membrane that has a pore size of 50 kilodaltons, being, big enough for the movement of smaller compounds like glucose or carbon dioxide.
It's kind of like the blood-brain barrier in a way, right?
Exactly, yeah. But small, too small for, mold or bacteria to get in. Viruses, they could get in as well, but they're much harder to actually input into the system.
All right, that is wild. So
yeah.
Well, there's so many questions that people have been asking about these things, but I suppose the first one on the chopping block would have to be like, where do you get the neurons from?
Yeah.
So do you like crack someone's skull open and just like yank them out?
Yeah, we hit somebody in the head, we bring them into the lab, and we don't do any of that stuff. Actually, initially in the early days of Cortical labs, we were actually using mice neurons for a lot of our research work. That was a little bit macabre as well, how we obtained the neurons, which was you would get a mouse pregnant, and in 10 days of its gestation, you would sacrifice the mouse, extract the pups, chop off the heads, pull out the brains.
Oh, okay, yeah.
So as you can tell, it's already not a very pleasant experience.
Not quite ethical harvesting at that point.
Yeah, I mean, it's common actually in the biomedical space for doing that, right?
But you're not doing that now, right?
We're not doing that now because, as we said, it's a little bit gruesome, quite dirty, and it's also very hard to maintain standardization, right? And we moved on from that into looking at using iPSC-derived neurons. So iPSC stands for induced pluripotent stem cells. So what's really interesting is that about 15 to 20 years ago, a researcher in Japan by the name of Professor Yamakana, who's since won the Nobel Prize in medicine, discovered that if you took any adult cell that has a nucleus, so red blood cells don't count because they don't have a nucleus, but white blood cells, skin cells, cheek cells, whatever, and you introduce 4 compounds known as Yamakana factors, you can potentially rewind the clock, the biological clock of any of these adult cells, back into an embryonic state, which means that they become stem cells that can morph into any type of cells if given the right compound or the gene expressions.
That is crazy. So you can literally take any cell in the body.
Correct.
Turn it into a stem cell, which used to be pretty controversial, right? As a cell type because of the harvesting.
Yes.
But now you can do that, ethically, effectively from anywhere on the body, and then turn it into that, and then you can turn the stem cells into anything. Like that's why stem cells are so cool, right? Because you can turn them into skin cells or neurons or whatever you want. Right?
And I think, if we go back historically, why there was a lot of controversy, particularly around stem cells, was that this was pre-Yamakana factors, right? We didn't know you could actually do this. And the source of most of our stem cells came from very pure mesenchymal stem cells, particularly, harvested from, newborn babies from the placenta and so forth, or even earlier where we were using throwaway IVF embryos. Now, that was very controversial, right? Because
I bet.
Yeah, right? Because, the question was, are you just creating embryos to get the stem cells? Was that ethical and not? And because you're creating a new entity, and it had no ability to make a consent, right?
Yeah, but that's not what we're doing now, right?
Correct, exactly.
It's quite different.
What we're doing now is very different because we're using adult pluripotent stem cells with particular focus on the adult part, because now we can actually get consent, and informed consent from the donor, whether they want to donate their cells for science, or right now in a lot of very interesting medical breakthroughs, people are now regrowing organs. And this has been, I guess, the holy grail of regenerative medicine, where you can take stem cells to regrow pancreatic islet cells, right? And
I think people that need organ transplants, for example, they don't have to wait on a list for someone to die
absolutely.
under the correct circumstances. They can actually grow these things in a
Correct.
lab. That's pretty cool.
And on top of that, what's more amazing about these organs is that they're actually you. So because they have the same genome, they have the same MHC major histocompatibility complex.
So there's less chance of rejection, for example.
There is zero chance of rejection, right? So
That's pretty cool.
it's very cool. Like you get the organs, you no longer need to go on anti rejection drugs, which mess up your immune system and so forth. So this is starting to really come through right now. So I think last year some very exciting news came out from China, where a patient with type 1 diabetes was actually treated with her own stem cells, and she started to produce insulin again.
Wow.
Yeah, so it was it was an actual cure. And then some interesting news came out as well, I think later part of last year, where several Japanese researchers started to notice that patients that had neural stem cells grown and re-injected back into their spinal cord, they all had like some level of paraplegia or quadriplegia, started to regain function again.
Okay.
Yes.
All right.
So this was very exciting for us in the field. I think it was the dream of Superman, right?
Yeah.
For one day that stem cells could actually be a thing.
Yeah.
So I think this is the testament of like innovation, and back to the whole quantum Hosky thing. You never know where technologies will take you, and I don't think Professor Yamanakana ever thought that his technology would actually lead to a computing platform.
Yeah, exactly. Because that's the other question that people always ask is like, where does it come from? Or kind of what is it? But also, what does it do? Like, what can you actually use this unit for? Like, are there any, like, I've seen, the example where you have this dish brain playing Pong against itself.
Yes.
I saw that you have this MNIST thing where, you recognize the hand-drawn numbers and stuff. Like, what you usually use artificial intelligence for.
Correct.
But is there anything like, that you think the dish brains might be more suited than regular AI for example?
Yeah, so I think the thing to try and understand here is, a lot of the stuff that we're trying to do with AI stems from the fact that things such as, language, or even earlier, like playing Go or Doom or whatever, are really hard to program with a lot of conditional if-else statements. And it was actually like feasibly impossible for a human to program a program like those. What machine learning and AI essentially did was it got the machine to program itself based on data, and to replicate a lot of the tasks that we humans take for granted. For us, you and I, we probably learned to speak at the age of 3, and we were conversing right now. You were generally intelligent. We can go make a cup of tea in an unknown kitchen. These are all the things that we're trying to get our machines to do, but we do it naturally, powered by our brains, right, which are composed of neurons. So if you boil it down to these like very logical sort of like abstractions, the things that we think about that are really, I guess, suited for dish brain or these kind of biological compute platforms are the fact that it's biological to begin with, which means that they also inherit the same drug profiles, diseases that we have. We have a paper that just came out in June where we showed that if you took cells that exhibited epilepsy seizures and put them on the platform, they recapitulate seizures on the dish brain, right?
So you can literally put it in here and it will have a seizure as a tiny little mini brain.
Correct.
And then you can effectively test out pharmaceuticals, right?
Exactly.
To see if it works or not.
That's what we did. We actually, to wind it back, the initial impetus of this was, we said, can we actually get any drug compound or any compound to have an effect? And our first experiment was actually, can we get it drunk?
Yes.
So we took the simplest compound that we could get in our laboratory, and every laboratory has this, is ethanol. Well, most of the time it's 80%, and nobody drinks 80%. So we had to water it down, and we started to titrate up the concentrations until we started to see really interesting results where the performance of the gameplay actually correlated with a downward trajectory as the concentrations raised.
Yeah.
So that was like the first indication. But we then followed it up with, the seizure form activity wave neurons that we then gave anti-epileptics. And we saw that, as we see in clinical practice as well, some people are resistant to some anti-epileptics, and some work better than others. Some of them have side effects and some of them don't. We were able to treat the seizure form activity and see it on the system when we applied these medications, but we also saw that depending on the type and the dosage, you got different performance in the gameplay in Pong. So I think that's one really exciting field that has immediate application. But the other application really is thinking about the fact that machine learning AI systems that we have today are hugely energy consumptive and data consumptive, right? A lot of the...
Because that's the issue, right? Like training one of these AI models is like, virtually the energy consumption of a mid-sized town for a year or
Absolutely.
something like that. It's huge.
Yes, it's huge. And on top of that, now inference is also being a real problem, right? As more people adopt the technology and use it. And this is really, I guess, one of the downsides of machine learning systems, right? Where a lot of it has to be pre-trained ahead of time because of the need for, say, backpropagating the error signals. What we've noticed with this system is that backpropagation doesn't seem to be happening. And that the learning is actually online. So what that means is that there isn't a training and inference phase. Training is part of the inferential loop.
Yeah.
What that actually lends really good application to things that operate in the real world with limited data sets. So things such as robotics, drones, metaverse gaming for NPCs, where you have to react in real time, cybersecurity.
What I heard as well is that this biological computing unit might be more suited for sort of complex data processing, like image processing and spatial reasoning, that kind of thing that traditional AI models are not great at.
Yeah, so I think that's probably a fair assertion as well, because a paper just came out I think with last week where we benchmarked, and this was actually shown at NeurIPS in 2022 in one of the posters in the reinforcement learning workshop, where we benchmarked the dish brain with the same game against 3 reinforcement learning agents. So DQN being the granddaddy of them all, you may know it as AlphaGo, and A2C and PPO, which are newer, more sample-efficient algorithms. And we saw that the biological system, if you sample constrain it, what I mean by that is that you're not giving more data, speeding up time and so forth. When you do a like-for-like comparison, the biological system outperforms significantly these reinforcement learning agents.
Okay.
So to your point, these are probably better suited for that kind of where you don't have that data sets. And one really important way to think about this, and Ilya Sutskever, who was the former CTO at OpenAI, he talked about data being like a fossil fuel, right? And that he reckons that the era of pre-training is over because we've exhausted that fossil fuel. We scraped everything off the internet. And the issue now is what is human, what is not, which goes back to the proof of human concept.
You kind of feed AI slop back into the AI, right?
Yeah, and then it's a well-known phenomenon. If you keep doing that, you end up reinforcing the hallucinations, reinforcing the error signals, and you end up with what's called model collapse.
Yeah.
And so, I think this is really interesting. If the analogy going back to the 1970 oil crisis, right? Places that had a lot of oil, like the United States, didn't really care so much because you could just dig more up. But places like Japan and Europe, which didn't have any oil and suffered tremendously from the embargoes and so forth, had to think of a better way to keep their motor industries going, and that was to make more efficient cars. And I think this is where we have to start thinking about AI systems, machine learning AI systems, where if you don't have the data sets, you don't have that fossil fuel, perhaps the way to go forward is to come up with more efficient systems.
Yeah, because the era of optimization was kind of put on halt a little bit because of Moore's law, effectively. Things were expanding was so cheap that you could just do it rather than optimize.
Absolutely.
Whereas back in the day, getting Super Mario Brothers coded, they had to do so much optimization to just get it to run.
100%. And
But yeah, I do think we have to wrap up here in a second. But I think the question that I do feel the need to round off on is this thing. Is it conscious?
That's a very controversial question, Trym. I think it really depends on. It's a little bit of a philosophy, and right now there isn't actually a real world consensus about what consciousness is. And I think this is something that we're working with the bioethics community. There's even a well-established nomenclature to agree on the terms, right? What is consciousness? What is sentience? Where do they start? Where do they end? What are the parameters? What are the objective measures? There are some people who believe in panpsychism, who believe that even a rock is somewhat conscious, right? And it's a spectrum. So perhaps, if you're a panpsychist, you probably think that this is somewhat conscious, but at a lower end of the scale. I certainly don't think that this is conscious because it's lacking the structures necessary for consciousness, right? Because we only see consciousness, I think, in higher ordered organisms that have very much more complex structures to their nervous system. So think of like octopi, mammals and birds kind of thing. This is quite equivalent to a flattened cockroach, and we don't really attribute much.
Like a worm almost. It's pretty small, right? Because how many neurons is in it?
So initially we started with 800,000 to 1 million, but we started to drop them down, and we're getting down to about 200,000 neurons and still getting quite good performance. I think this is the difference between engineering and science. For us as scientists, it's not enough to just show an effect and try to grow it up. What we're trying to do now is to decimate and try to find what is the smallest unit that actually still has any effect, because that removes the confounding factors, and we can start experimenting with perturbing the systems and understanding better how these things are working.
Yeah, and building them up and connecting them in different ways. But anyway, it's been a pleasure to have you both, here at the booth and obviously in the podcast.
Yes.
So, by the way, if people are interested, how can they reach you guys?
Yeah, you can reach us out on our website, corticallabs.com. We have a very active Discord community as well. I'm available also on social media, so hit me up on the Discord or reach out to me on Twitter or X, whatever it's called now. And stay tuned to Quantum Hosky, because I think they're planning to use some of this stuff as well in some of their work. But I think a big shout out to the community as well. I think there's something Trym was asking me, what do I want to impart with the viewers and listeners, and I think it's that you guys have a significant power out there to influence the pace and shape of humanity, right? In progress, because if you think about the amount of excitement, the amount of adoption of new technology, the futurism in the blockchain community, and also the financial power that you guys have, a lot of scientists out there have really great ideas and just don't have the ability to get funded. Things and projects such as decentralized science, DeSci, are actually really exciting, I think, as a path forward to accelerate the pace of progress. And if you want to see more things like DishBrain come out or more quantum technologies, this is, I think, the platform to hit it up.
Yeah, and there's so much in the space as well that you should check out related to that. For example, Catalyst as a decentralized funding platform.
Yes.
That's a whole other conversation. So, I think we'll leave it there for today. But thank you very much for joining me.
Thank you.
Thank you for watching.